In the last video, I said it's important that influenza is a negative-stranded RNA virus, and also it's important that it's segmented. But we didn't get to why those things are important, so let's do that now. So you probably know that influenza causes seasonal outbreaks and occasionally widespread pandemics. And every single year, the WHO and the FDA have to develop a new influenza vaccine prior to the outbreak to help prevent influenza illness. And that's different from other vaccines that are given at a few specified times throughout someone's life and provide long-lasting immunity. So the question is, why is influenza different? Well, first we briefly mentioned that influenza encodes its own RNA-dependent RNA polymerase. And it turns out that in general, this RNA polymerase is really prone to making errors. And that results in random point mutations in hemagglutinin and neuraminidase proteins in addition to all the others. And if enough mutations occur so that our antibodies no longer recognize the hemagglutinin and the neuraminidase, then a new subtype of influenza has been created. And this concept is called antigenic drift. It's not a huge change, it's the virus just gradually drifting from one subtype to another as it accumulates these point mutations. And both influenza A and B can undergo antigenic drift, although influenza B does so less rapidly than influenza A. So this antigenic drift is what causes the new seasonal strain every year that requires a new seasonal vaccine. But this is different from antigenic shift. And shift is where the influenza genome being segmented comes into play. So how does antigenic shift occur? Well, normally when a cell is infected with the flu, 8 gene segments come in, are copied, and then the same 8 segments are packaged into a new viral particle. But if a single cell, whether it's animal or human or whatever, is infected with 2 different influenza viruses, the 2 viruses can actually exchange RNA gene segments that leads to the creation of a third virus that's different from both of the parent viruses. And this exchange of genes is called reassortment. You might have noticed that people refer to influenza in a funny way. Sometimes they'll say H1N1 or H3N2. Well, that means the flu strain has a certain hemagglutinin protein and a certain neuraminidase protein. So with reassortment, to give you an example, if one cell is infected with both H1N1 and H3N2 influenza, you could theoretically produce a new H3N1 influenza strain. So antigenic shift can result in pandemics, which are a global spread of a new and totally different influenza strain to which the population has little to no immunity, and so it can spread very easily from human to human. But one thing that's interesting is that this antigenic shift only happens with influenza A and not B. So why is that? The reason it turns out is that influenza A can infect animals, all kinds including birds, pigs, horses, and more. And what does that have to do with it? Well, when viruses infect other species, they adapt to those species, and that gives them a lot more genetic diversity. So when reassortment happens, including these non-human strains, it can produce new viruses that by chance are infectious to humans, but are very different from what humans are used to seeing because the building blocks came from animals. And as an example, do you remember the swine flu? So that was the most recent pandemic in 2009, and that was a reassortment of avian, swine, and human viruses. And that reassortment event that we talked about happened in a pig, and that's why it was called swine flu. And that could never have happened with influenza B, which does not infect animals.